Okay, let's just jump right in. Every single day, you come into contact with thousands of chemicals. They're in our food, the air, the water, the products we use. Many are foreign to our bodies, what scientists call xenobiotics.
Right.
And this isn't exactly a new thing. Life on Earth, especially animals, has been encountering these substances for, well, millennia.
Absolutely. Think about all the complex chemicals plants produce: pigments, those defensive compounds like phytoalexins.
Yeah, and outright toxins like the lethal compounds found in poisonous mushrooms.
Right.
You know, things like amanitin or gyrometrin.
No.
Nasty stuff.
That's right. And the ability to process and eliminate these foreign chemicals isn't just a metabolic footnote.
Right.
It's been absolutely fundamental to adaptation, to survival, really, for animal life throughout evolution.
Without a pretty sophisticated system to handle them, they would just, build up, accumulate in tissues.
Exactly. And cause significant harm.
And that's the core of what we're diving into today. We've got some foundational material that lays out this incredible defense system our bodies have evolved. Our goal here is to go deep, to really unpack every concept and hopefully gain a truly mastery-level insight into how our bodies handle these chemical invaders, especially drugs.
Because drugs, as it turns out, are just a specific type of xenobiotic.
Right. The same machinery that handles a compound from a plant also handles your medication. It's all the same system.
And as we explore this, what quickly becomes apparent is the sheer complexity. We were talking about a vast array of diverse enzymes, crucial interactions between them, and really importantly, significant individual differences that dictate how any one person responds to a particular drug or environmental exposure.
So to really master this, we need to understand all those moving parts, the enzymes, the interactions, the variability.
That's the goal.
So let's start with that fundamental challenge. Constant exposure to foreign chemicals. As you mentioned, plants are a major source, just full of these diverse compounds. And those poisonous mushrooms are a stark reminder of the potency some natural xenobiotics can have. Amanitin, gyrometrin, orolanin, muscarine, ibotenic acid, muscimol, psilocybin, coprine. I mean, a truly nasty lineup of toxins.
A real rogues gallery.
Yeah.
And animals needed to somehow survive exposure to those.
So for animals to live in environments where they might ingest or encounter these chemicals, developing robust metabolic capabilities it was under strong evolutionary pressure.
Absolutely. This ability is critical for survival and adaptation.
The key players in this system are the enzymes. Historically, they were called drug metabolizing enzymes, especially by pharmacologists, right?
That was a common term, yes.
But that term feels a bit too narrow now because they handle far more than just pharmaceutical.
Much more. Calling them xenobiotic metabolizing enzymes is much more accurate. It really is a better description.
Okay.
Over evolutionary time, a myriad of these diverse enzymes has evolved across different animal species. And you see marked variations between species.
Well, why is that? Different diets.
That's likely the main driver. Reflecting the different dietary and environmental challenges they faced during their evolution, a koala's metabolism is vastly different from a fox's because their diets are just so specialized.
Right, and it's not just chemicals from outside the body, is it? These same enzyme systems also evolved to help process and eliminate certain endogenous chemicals, things our own bodies make.
Precisely. Substances our bodies produce that could become harmful if they built up, like bilirubin, steroid hormones, or catecholamines.
So the same enzymes, or very similar ones, handle both jobs, foreign chemicals and potentially problematic internal ones.
Often, yes. The same or closely related enzymes handle both. This dual function just underscores their central role in maintaining health at the cellular and organism level.
Which leads us directly to drugs. Any medication you take is, by definition, a xenobiotic.
Absolutely. And your body's ability to metabolize and clear that drug relies entirely on the same enzymatic pathways and transport systems that handle all the other chemicals you encounter.
It's a fascinating historical link, too. Many modern drugs, even current cancer treatments, were derived from plants. And numerous drug discoveries stem directly from investigating traditional or folkloric claims about plant-based medicines, based on centuries of just observing how these natural compounds affect people.
It highlights that deep evolutionary connection. Our bodies have been interacting with these kinds of molecules for a very long time.
Now, here's an interesting paradox, maybe. This metabolic system is overwhelmingly beneficial because it lets us eliminate foreign chemicals, prevents them from building up to toxic levels.
There's a but. That very process creates significant challenges, particularly in the complex world of drug development and clinical medicine.
Like what kind of challenges?
Well, first, predicting human drug behavior based on animal studies is tricky because of those species differences in enzyme expression we just talked about.
Okay.
Then there's the huge variability in metabolic capacity between individual humans. That's a big one.
Right, why one person responds differently than another.
Exactly. Plus, drug interactions are a major concern, where different drugs compete for the same enzymes.
And sometimes the metabolic process itself can accidentally convert a chemical into something even more toxic or carcinogenic than the original substance. Is that right?
That can happen too. So while it's our primary defense, the intermediate products can sometimes be problematic. Metabolic activation of protoxins.
And it's worth remembering that today's xenobiotics go far beyond just plant compounds.
Oh, absolutely. Environmental pollutants, food additives, cosmetic ingredients, agrochemicals, processed food components, and of course, pharmaceuticals. It's a complex chemical world.
All of which brings us to the fundamental chemical problem these enzymes are designed to solve: hydrophobicity. Most xenobiotics are hydrophobic, meaning they're lipid soluble.
Right. And if they remained in that state, they would readily dissolve and just accumulate in the lipid compartments of our bodies. Think fat tissue, the phospholipid membranes of our cells.
And that accumulation is the root cause of potential toxicity for many substances.
It is. Because hydrophobic drugs can just slip across cell membranes to get into cells in the bloodstream. But once they're embedded in those fatty environments, they're very difficult for the body to excrete in watery fluids like urine.
Stuck in the membrane, basically.
Pretty much. So the core job, the fundamental task of these metabolic enzymes is to transform these hydrophobic, lipid-soluble chemicals into derivatives that are much more hydrophilic, more water-soluble.
And once they're water-soluble,
Much easier to eliminate. Hydrophilic compounds dissolve readily in aqueous body fluids like blood and urine. They can be efficiently excreted either by the kidneys into the urine or for larger molecules transported into the bile by the liver and eliminated via the intestine and feces.
Okay, this is where we talk about the two phases of drug metabolism, traditionally categorized by the type of chemical reaction involved.
Right, phase 1 and phase 2. Phase 1 reactions typically involve oxidation, reduction, or hydrolysis. What these reactions usually do is introduce or expose a reactive functional group on the molecule.
Things like OH groups or carboxyl groups.
Exactly. A hydroxyl, OH, carboxyl, TCOOH, self-hydryl NSH, ether O, or really NH2 group, things like that.
So adding a little chemical handle almost.
That's a good way to think of it. Now, while adding these groups does slightly increase water solubility, that's not usually their main effect in phase 1.
Okay.
Phase 1 reactions often dramatically change the chemical structure and therefore the biological properties of the substance.
And usually this leads to drug inactivation. That's the goal.
That's the most common outcome, yes. The modified molecule is less able to bind to its biological target. However, there's a very important exception: metabolic activation.
This happens with prodrugs, right? Drugs that are inactive until they're metabolized.
Exactly. They're designed that way. A phase 1 reaction, like hydrolysis of an ester or ammonia linkage in a prodrug, can release the active compound. Sure. Anti-tumor drugs like cyclophosphamide or antithrombotic drugs like clopidogrel, they require activation, often by specific CYP enzymes. But prodrugs can also be activated by enzymes in the gut, blood, or inside cells. FMOs or other hydrolytic enzymes can do it too. It's actually a pretty clever pharmaceutical design strategy to improve drug stability or delivery.
Okay, so phase 1 adds functional groups. Usually inactivates, but sometimes activates. What about phase 2?
Phase 2 reactions are called conjugation reactions. Here, an enzyme catalyzes the attachment, or conjugation, of a second molecule to the substrate.
And the second molecule is usually something small and water-soluble from the body.
Precisely. Often a small, highly water-soluble endogenous compound like glucuronic acid or sulfate.
And this attachment often happens at one of those functional groups introduced back in phase 1.
Yes, frequently. That functional group acts as the attachment point, although some drugs can undergo phase 2 conjugation directly without needing phase 1 first.
And this is where you get the big increase in water solubility.
This is the key step for that, yes. The resulting conjugate product is much more hydrophilic.
Which helps it get eliminated.
Exactly. This improved water solubility is crucial for facilitating transport out of cells and tissues, often via specific efflux transporters, and then excretion from the body.
So phase 2 generally serves two purposes: facilitating elimination.
And often inactivating potentially reactive or toxic metabolites that might have been generated during phase 1 oxidation. It's kind of a cleanup and prepare for exit step.
It sounds like these enzymes are organized into families with shared ancestry, this idea of superfamilies.
They are. In the mammalian genome, enzymes and receptors that are evolutionarily related are grouped into superfamilies based on similarities in their structure and function.
And within those are subfamilies.
Right. Subfamilies are more closely related, often encoded by genes found close together on a chromosome. The xenobiotic metabolizing enzymes are prime examples of this organization.
So for phase 1 oxidation, the main superfamilies are the CYPs, FMOs, and epoxide hydrolases. CYPs and FMOs are mentioned specifically as superfamilies or subfamilies.
Correct. And for phase 2 conjugation, we have several superfamilies of transferases. The key ones we'll really dig into are the glutathione S transferases, or GSTs.
Okay.
UGTs, which are the glucuronosyl transferases, sulfotransferases, or SULTs, and acetyltransferases, NATs, and methyltransferases, the MTs. Each type catalyzes the addition of a different kind of molecule.
And for phase 2 to happen, the substrate generally needs to have an oxygen, nitrogen, or sulfur atom somewhere on the molecule to act as the attachment site.
Yes, they need an acceptor site for those hydrophilic moieties like glutathione, glucuronic acid, sulfate, or an acetyl group. Think again about a drug like phenytoin.
Okay.
After a CYP in phase 1 as a hydroxyl group, that OH site is a perfect acceptor for a UGT enzyme in phase 2 to attach glucuronic acid.
Right.
The resulting phenytoin glucuronide is highly water-soluble and basically tagged for elimination.
And the overall biological effect is usually to convert the drug into a form that can't bind its target receptor anymore, dampening its effect.
That's the standard outcome. It's all about preparing the molecule for removal from the body and stopping its action.
So where does all this enzymatic action actually happen in the body? Is it everywhere?
Well, these enzymes are found in most tissues, but the highest levels are definitely concentrated in the gastrointestinal tract, especially the liver, but also the small and large intestine play really big roles.
The small intestine too. Why there?
Well, think about it. Anything you take orally is first exposed to your gut bacteria, which can actually metabolize some compounds even before absorption.
Okay.
And then, as the drug is absorbed across the gut wall, it passes through the epithelial cells lining the intestine. And those cells are packed with these metabolic enzymes. It's a critical initial site of metabolism.
A first line of defense, sort of.
Exactly. But the liver is truly the major metabolic clearinghouse for both endogenous compounds and xenobiotics.
Right, because blood from the GI tract goes straight to the liver through the portal vein.
Before it enters the general circulation, this is the famous first pass effect. The liver can extensively metabolize drugs on this initial pass.
And even after that first pass, the drug keeps circulating back through the liver.
Yep. With each subsequent pass through the liver, it continues to metabolize the drug until it's eventually eliminated.
So drugs that aren't significantly metabolized during this first pass or subsequent passes will just stay in the body longer, right? They'll have longer half-lives.
Yes, that's a key factor in pharmacokinetics. This is why, in drug development, compounds are often designed aiming for a half-life of around 24 hours. That allows for convenient once-daily dosing.
Makes sense. If a drug is poorly metabolized, managing its levels could be trickier.
Much trickier. And while the liver and gut are key, other tissues also contribute significantly to metabolism. Think about the nasal mucosa and lungs, really important for substances you inhale or that are administered as aerosols.
And within the cells themselves, where do these enzymes physically reside? Where do they hang out?
There's a specific cellular localization. Many of the key phase 1 enzymes, including the CYPs, FMOs, and epoxide hydrolases, along with some important phase 2 enzymes like the UGTs, are actually embedded within the membrane of the endoplasmic reticulum, the ER.
The ER, that vast network of lipid bilayers inside the cell.
That's the one.
Which makes perfect sense for handling hydrophobic molecules, right? They can enter the cell, easily slip into the lipid membrane of the ER.
And right there are the enzymes waiting to modify them. It's an ideal setup.
So a hydrophobic substrate embeds in the ER membrane, gets oxidized by a CYP maybe?
And the resulting product could potentially be directly conjugated right there by UGTs that are also located in the ER membrane.
Or it could move elsewhere.
Or the modified molecule might move out into the cytosol, the main cell fluid, to be handled by cytosolic enzymes like most of the other phase. Phase 2 transferases, the GSTs, SLTs are mostly cytosolic.
Okay. And after all this metabolism happens, say in the liver cells, the resulting metabolites need to be transported out of the cell.
Right. They are moved across the hepatocyte's outer plasma membrane. They can go either into the bloodstream, heading towards the kidneys for elimination in urine,
Oh,
or, especially for larger metabolites, they can be actively transported across a different part of the membrane into the bile canalicculus. That leads into the bile ducts and ultimately elimination via the intestine and feces.
does size matter here? Which way they go?
It often does. Higher molecular weight compounds tend to be preferentially excreted into bile, but it also depends heavily on the specificity of the various membrane transporter proteins that actually move the molecules across the membranes.
Okay, let's really dig into those phase 1 reactions now, starting with the real heavy hitters, the CYPs, the cytochrome P450 superfamily.
Right, CYPs, they're absolutely central players. Each enzyme contains a heme molecule.
Like in hemoglobin, that iron structure.
Very similar iron protoporphyrin IX structure, yes. It's non-covalently bound in the CYP's active site. And it's this heme iron that binds oxygen, which is essential for the oxidation reaction.
So they use oxygen to chemically change the substrate.
Exactly. They utilize molecular oxygen, O2, and hydrogen ions, which are ultimately supplied by a cofactor called NADPH. This electron transfer is facilitated by another enzyme partner called NADPH-CYP oxidorreductase.
And the reaction itself.
The core reaction basically inserts one atom from the O2 molecule into the substrate, modifying it, and the other oxygen atom ends up forming a water molecule. Substrate plus O2 plus NADPH plus a proton gives you oxidized substrate, water, and NADP plus oxidate.
The material also mentions something about uncoupling in CYPs. What's that about?
Yes. The process isn't always perfectly efficient. Sometimes a CYP enzyme can go through its catalytic cycle, consume oxygen and NADPH, but without actually oxidizing the intended substrate molecule. This is called uncoupling.
And that leads to.
It can lead to the generation of activated oxygen species, like superoxide radicals. These are reactive oxygen species, or ROS.
Which can be damaging to cells, right? Oxidative stress.
They certainly can be. Normally, our cells have antioxidant defenses, like the enzyme superoxide dismutase, to handle these ROS. But if ROS production becomes excessive due to high CYP activity or uncoupling, it can overwhelm these defenses, leading to oxidative stress. And chronic oxidative stress can disrupt cell function and is implicated in various diseases, including things like hepatic cirrhosis.
Got it. Structure, reaction, potential side effect. And where are these CYPs mainly located within the cell again?
They're primarily embedded in the ER membrane. Most of the enzyme protein actually faces the cytosol, the main cell compartment. And interestingly, a single NADPH-based CYP oxidorreductase enzyme seems to service or provide electrons to many different CYP enzymes within that ER membrane.
Now, about their substrate specificity, there are 57 human CYPs identified. And they catalyze an incredible range of reactions, removing alkyl groups, adding hydroxyl groups, oxidizing nitrogen or sulfur, moving acylamine groups, even removing halogens.
It's a large and versatile family, absolutely.
Yeah.
But it's really important to distinguish between them. Not all 57 are involved in metabolizing foreign compounds, xenobiotics.
Oh, okay. What do the others do?
Some are highly specialized and dedicated to synthesizing crucial endogenous molecules, things like steroid hormones, fatty acids, or bile acids. These particular CYPs usually have very tight, very specific substrate preferences.
For example?
For instance, the enzyme CYP19, which is also called aromatase, its job is specifically to convert testosterone and androstenedione into estrogens. It doesn't really touch foreign chemicals, and we actually have specific drugs designed to inhibit just that enzyme for certain therapies.
Okay, so some CYPs are highly specific for internal jobs, but the ones that handle xenobiotics, the material describes them as promiscuous. That's quite a term.
It is, but it's actually pretty accurate in a biochemical sense. These xenobiotic metabolizing CYPs can bind and metabolize a wide variety of structurally diverse substrates. They aren't locked into just one specific molecule.
And this promiscuity, combined with the fact that there are multiple different CYPs, is what allows our bodies to process the vast universe of chemicals we encounter.
That's exactly it. It provides broad coverage. A single drug or chemical might even be a substrate for several different CYP enzymes, or it might be metabolized at multiple different positions on the molecule by the same or different CYPs.
But this promiscuity comes at a cost. The material suggests they seem to sacrifice catalytic speed for this broad specificity.
That's generally true. Compared to enzymes involved in rapid intermediary metabolism, like glycolysis, CYPs metabolize their substrates relatively slowly.
So that's why drug half-lives are often hours or days, not seconds.
It's a major factor. Endogenous compounds, handled by faster enzymes, might have half-lives measured in seconds or minutes. while drugs metabolized by CYPs often have half-lives ranging from, say, 3 to 30 hours or even longer. But this level of activity is still more than sufficient to handle the concentrations of drugs we typically encounter.
This promiscuity, though, it seems like it could lead to real problems when you take multiple drugs at the same time.
It's a huge issue in medicine and the leading cause of adverse drug reactions, or ADRs.
How does that happen?
Well, if you take 2 or more drugs that are both substrates for the same CYP enzyme, they will essentially compete for access to that enzyme's active site.
Like too many people trying to get through one door.
Good analogy. This competition can inhibit the metabolism of one or both of the drugs.
Leading to higher than expected levels of the drug in the bloodstream.
Exactly. And this is particularly dangerous with drugs that have a narrow therapeutic index, where the dose needed for effect is very close to the dose that causes toxicity. Even a relatively small increase in concentration due to inhibited metabolism can push the drug into the toxic range.
Which is why drug labels often warn about interactions.
Precisely. They have to list potential interactions and often specify the CYPs involved. And in drug development, preclinical studies absolutely must consider potential interactions with existing medications that might share metabolic pathways.
Can you give an example?
Sure. Combining certain cholesterol-lowering statins with certain antidiabetic drugs, if they happen to be metabolized by the same CYP, might require careful dose adjustment, or even choosing alternative drugs to avoid dangerous interactions.
How are these CYPs named? There are so many, it must get confusing.
There's a standard nomenclature, thankfully. It starts with CYP, followed by a number for the gene family, then a letter for the subfamily, and finally a number for the specific gene or enzyme within that subfamily.
So CYP3A4 means?
Family 3, subfamily A, gene number 4. It helps keep track.
And out of those 57 CYPs, it sounds like only a handful are responsible for metabolizing the vast majority of drugs we use.
Yes, that's correct. A relatively limited number, primarily from families 1, 2, and 3, account for the bulk of xenobiotic metabolism in humans. We repeatedly see about 12 mentioned as being particularly important. CYP 1A1, 1A2, 1B1, 2A6, 2B6, 2C8, 2C9, 2C19, 2D6, 2E1, 3A4, and 3A5.
And these handle not just drugs, but also many compounds from our diet and the environment.
Correct. They're broadly involved.
And while the liver has the highest concentration, ensuring that crucial first pass effect, they're also found in other places like the gut, lungs, kidneys, even the brain, just at lower levels.
That's right. Now, within that group, the ones most active in metabolizing therapeutic drugs are generally considered to be the CYP2C, 2D, and 3A subfamilies.
And CYP3A4 is the real giant here.
It really is. CYP3A4 is the most abundant CYP enzyme in the human liver, and it's estimated to metabolize well over 50% of all clinically used drugs. It's incredibly important.
Wow, over half for just one enzyme. That's huge.
It is. Now, some of the other CYPs we mentioned, like those in the 1A, 1B, 2A, 2B, and 2E families, they might be less critical for metabolizing therapeutic drugs themselves, but they play a significant, sometimes detrimental role.
How so?
They're often involved in activating protoxins and procarcinogens, inactive chemicals from our diet or environment that become toxic or cancer-causing only after being metabolized by these specific CYPs into reactive metabolites.
So they can turn harmless things harmful sometimes?
Unfortunately, yes. That metabolic activation is a key concern.
You mentioned individual differences earlier. How do CYPs factor into why people respond so differently to the same drug?
This is where CYP polymorphism comes in. It's a major factor. There are very large differences in the expression levels and, consequently, the activity of CYP enzymes between individuals.
What causes these differences?
It's largely down to 2 things. Genetic polymorphisms, meaning common variations in the DNA sequence of the CYP genes themselves, and also differences in how the expression of these genes is regulated in different people.
And several key CYP genes are known to be highly polymorphic, lots of common variations.
Yes, very much so. Genes like CYP2A6, 2C9, 2C19, and especially CYP2D6 are known to have many common allelic variants that affect enzyme function.
And these genetic variations contribute significantly to that variable drug metabolism we see between people.
Absolutely. They are major determinants of variable drug response and susceptibility to ADRs. And we even see marked ethnic differences in the frequency of these polymorphisms.
Like what?
Well, for example, about 10% of Caucasians have genetic variations that lead to a lack of functional CYP2D6 enzyme activity. And for CYP2C19, it's even higher in some Asian populations, maybe up to 25% lacking functional enzyme.
And this has real consequences in the clinic, right? It's not just theoretical.
Oh, absolutely. It has led to drugs being withdrawn from the market or requiring extreme caution and specific dose adjustments based on genotype. The CYP2D6 polymorphism is a classic example.
How does that manifest?
Well, take codeine, a common pain reliever.
Okay. Isn't codeine a prodrug? It needs to be metabolized to work.
Exactly. Codeine itself has little analgesic effect. It needs to be O-demethylated, have a methyl group removed to form morphine, and it's the morphine that actually provides the pain relief.
And that conversion is done by?
Primarily by CYP2D6. So if an individual has genetic polymorphisms that result in inactive or completely absent CYP2D6 enzyme.
They won't make much morphine from the codeine.
Right. They'll convert little or no codeine to morphine and therefore get very little or sometimes absolutely no analgesic effect from a standard dose of codeine. They're often called poor metabolizers.
Wow. Are the really major CYPs like 3A4 also highly polymorphic in this way?
Well, allelic variants for enzymes like CYP1B1 and CYP3A4 do exist, but they seem to be less common, or their functional impact might be less pronounced compared to the variations seen in 2D6 or 2C19. They don't seem to explain the bulk of the typical inter-individual variability we see for 3A4 activity, for instance. However, rare homozygous mutations in CYP1B1 are associated with a specific condition called primary congenital glaucoma.
Okay. Going back to drug interactions, besides competition for the enzyme's active site, the material also discusses something called CYP inducers. What are those?
This is another critical mechanism driving variability in interactions. Some xenobiotics, or drugs, don't inhibit CYPs. They actually increase the amount of CYP enzyme produced.
How do they do that?
They typically work by activating specific nuclear receptors proteins inside the cell that act as transcription factors. When activated, these receptors increase the transcription of the target CYP genes, leading to higher levels of the CYP enzyme being made.
And what's the consequence of having more enzyme?
Well, it can increase the metabolism of the inducer drug itself. That's called autoinduction. This can lead to the drug's own levels decreasing over time, potentially causing a loss of efficacy.
Or it could affect other drugs.
Yes, and that's often the bigger concern clinically. Inducing a CYP can significantly increase the metabolism of other co-administered drugs that happen to be substrates for that particular induced enzyme. This speeds up their clearance and lowers their plasma levels.
Are there common examples of inducers, like certain drugs or supplements?
Yes, definitely. Certain steroid hormones can act as inducers, and the herbal supplement St. John's wort is a very well-known potent inducer of hepatic CYP3A4.
Ah, St. John's wort. I've heard about interactions with that.
It's a classic case. Taking St. John's wort can dramatically increase the metabolism of many oral drugs that are substrates for CYP3A4. The most famous example is probably with oral contraceptives. It can lower the steroid levels so much that it leads to contraceptive failure.
Wow. And diet can influence this too. Inhibitors and inducers in food.
Absolutely. Certain foods contain natural compounds that act as either CYP inhibitors or inducers. Grapefruit juice is the textbook example of an inhibitor.
Right, grapefruit juice. What does it do?
It contains compounds, particularly furanocoumarins and possibly naringin, that are potent inhibitors of CYP3A4, specifically in the intestinal wall.
So it blocks the metabolism of drugs right there in the gut, even before they get absorbed properly or reach the liver.
Exactly. It essentially increases the drug's bioavailability by preventing that initial gut wall metabolism. This can significantly increase the plasma levels of many drugs that are CYP3A4 substrates.
What kind of drugs are affected?
Numerous classes, some antihypertensives, immunosuppressants, antidepressants, antihistamines, statins. The list is quite long. This is why you often see specific warnings on drug labels about avoiding grapefruit juice.
And the story of the antihistamine terfenadine really illustrates just how serious this inhibition can be.
It's a stark reminder, yes. Terfenadine, brand name Seldane, was an antihistamine that was ultimately withdrawn from the market.
Why? What happened?
Terfenadine itself was actually a prodrug. It was metabolized by CYP3A4 to its active form, fexofenadine, which is now sold as Allegra. Fexofenadine was effective and safe.
Okay.
However, if CYP3A4 activity was inhibited, say, by taking it with certain other drugs like the antibiotic erythromycin, or even just by drinking grapefruit juice, the metabolism to fexofenadine was blocked.
So the parent drug terfenadine built up.
Exactly. The parent drug, terfenadine, would accumulate to high levels in the body. And it turned out that terfenadine itself, at these high concentrations, was cardiotoxic. It could cause a dangerous, potentially fatal type of heart rhythm disturbance called torsades de pointe, a form of ventricular tachycardia.
So the problem wasn't the active drug, it was the original drug accumulating because its metabolism was blocked.
Precisely.
Yeah.
The active metabolite, fexofenadine, is not cardiotoxic and was subsequently developed and marketed as a safe alternative replacement. It was a direct outcome of understanding this metabolic pathway and its vulnerability to inhibition.
A powerful lesson. Okay, moving on from the CYPs, what about the other phase 1 oxidation enzymes mentioned, the flavin-containing monoxygenases or FMOs?
Right, the FMOs. They're another superfamily of phase 1 enzymes involved in oxidation. Like many CYPs, they're found at high levels in the liver and are also bound to the ER membrane, which favors their interaction with hydrophobic substrates.
They're one main FMO.
FMO3 is the most abundant form in the human liver. It's known to metabolize various compounds, including nicotine, some H2 receptor antagonists like cimetidine and ranitidine, antipsychotics like clozapine, and certain antiemetics like etopride.
And they are linked to that rare genetic condition, fish odor syndrome?
Yes, trimethylaminuria. That syndrome is caused by a rare genetic deficiency in the FMO3 enzyme. It prevents the metabolism of trimethylamine, TMA, which is derived from dietary precursors like choline and carnitine, often found in foods like fish, eggs, and liver.
So without FMO3,
unmetabolized TMA builds up in the body and is excreted through sweat, breath, and urine, causing a strong, socially distressing fish-like odor. The condition can usually be managed by limiting dietary intake of TMA precursors.
compared to the CYPs, the material suggests FMOs are relatively minor contributors to overall drug metabolism.
Generally considered true, yes. They handle fewer drugs than the major CYPs. And importantly, FMOs almost always produce benign, non-reactive metabolites. They don't typically create those electrophilic intermediates we worry about with some CYP reactions.
And crucially, they aren't involved in the same kind of drug interactions.
That's a major difference. FMOs are not readily inhibited or induced by the nuclear receptors and other mechanisms that cause so many CYP-mediated drug interactions. This means FMOs are not typically expected to be involved in significant drug interactions via inhibition or induction pathways.
So theoretically, a drug primarily metabolized by an FMO might have a more predictable pharmacokinetic profile when taken with other medications.
That's the potential advantage, yes. The material contrasts etopride, which is primarily metabolized by FMO3, with an older drug called cisapride, which was metabolized by CYP3A4 and had significant interaction issues. Etopride is considered much less likely to cause such interactions.
Interesting. So designing new drugs to be FMO substrates could be a strategy to avoid interaction problems.
It's a potential strategy being considered in drug design, yes.
Yeah.
Introducing structural elements that favor FMO oxidation could lead to more predictable metabolism and pharmacokinetics.
Okay. What about the third group of phase 1 enzymes? Mention the hydrolytic enzymes.
These are enzymes that break chemical bonds by adding a molecule of water that's hydrolysis. The key players here that we need to discuss are the epoxide hydrolysis, EHs, and the carboxyl esterases.
Let's start with epoxide hydrolysis, EHs.
Right. EHs, as the name suggests, hydrolyze epoxides. Epoxides are these three-membered ring structures containing an oxygen atom. They're often formed as intermediate metabolites, particularly during oxidation reactions carried out by CYPs.
And epoxides are problematic.
They can be highly problematic. Epoxides are often highly reactive electrophiles. They have a strong tendency to react with and covalently bind to cellular nucleophiles, critical molecules like proteins, RNA, and DNA. This binding can cause cellular toxicity, mutations, and potentially lead to cancer initiation.
So EHs are basically detoxifiers for these reactive intermediates.
Exactly. They play a crucial detoxification role by adding water across the epoxide ring, opening it up to form less reactive diols, compounds with two hydroxyl groups.
There are two main forms mentioned, soluble EH, SCH in the cytosol, and microsomal EH, MEH in the ER membrane.
Correct. They have different locations and somewhat different substrate preferences. MEH, the microsomal form, is particularly involved in metabolizing epoxides formed on cyclic or aromatic systems.
Like with the drug carbamazepine.
Yes, that's a good example. Carbamazepine, an anti-epileptic drug, is metabolized by CYP3A4 to an active epoxide metabolite. This epoxide is then hydrolyzed and inactivated by MEH to form an inactive dihydrodyl metabolite.
So if MEH activity is inhibited, the levels of that active epoxide metabolite could potentially increase.
Precisely. Certain other drugs, like valnoctamide or valproic acid, can inhibit MEH. If given together with carbamazepine, this inhibition can lead to elevated levels of the active carbamazepine epoxide, potentially causing increased side effects or toxicity. This highlights the importance of this enzyme pathway and has actually influenced the design of some newer anti-epileptic drugs to specifically avoid metabolism involving epoxide intermediates.
Okay, and what about the soluble SEH? It seems to be more focused on metabolizing fatty acid epoxides.
Yes, that's its main role. SEH metabolizes epoxides derived from endogenous polyunsaturated fatty acids like arachidonic acid and docosahexaenoic acid, DHA. These fatty acid epoxides, sometimes called ETs or EDPs, are actually important signaling molecules themselves.
Signaling molecules, what do they do?
They are part of the complex arachidonic acid signaling cascade and generally have beneficial effects that can help resolve inflammation, lower blood pressure, and mediate pain relief. They act as vasodilators and anti-inflammatory mediators.
But SEH breaks them down quickly.
It does. SEH hydrolyzes them fairly rapidly into their corresponding but generally less biologically active diols.
And this has led to therapeutic interest in inhibiting SEH.
Exactly. The idea is that by blocking SEH with specific inhibitors, you can increase the levels and prolong the action of these beneficial fatty acid epoxides. This strategy has shown promise in preclinical models for reducing inflammatory conditions, neuropathic pain, and even potentially lowering blood pressure. It might also synergize with NSAIDs.
So EHS detoxify harmful epoxides generated by CYPs, but inhibiting a specific SEH, might be a therapeutic strategy to increase levels of beneficial signaling metabolites. That's a fascinating duality.
It really is, and it just shows the intricate balance and complexity within these metabolic pathways. Sometimes you want to speed up metabolism, sometimes you might want to slow it down for a specific target.
Okay. What about the other hydrolytic enzymes, the carboxylesterases?
Right, CESs. This is another superfamily of hydrolytic enzymes, found in both the ER and the cytosol in many different cell types. As their name implies, they catalyze the hydrolysis of chemicals containing ester or amide linkages.
And they can function in detoxification or activation?
Both. They can detoxify certain compounds by breaking them down, but importantly, they are often involved in activating prodrugs. Many prodrugs are designed with ester or amylamide linkages that get cleaved by carboxylesterases inside the body to release the active free acid or aminamide form of the drug.
Like the cancer drug irinotecan we keep mentioning.
Yes, irinotecan is a classic example. It's a prodrug that gets converted primarily by the intracellular carboxylesterase CES2 into its highly potent active metabolite SN38, which is the actual topoisomerase inhibitor that kills cancer cells.
And unlike many CYPs, are carboxylesterases generally involved in significant drug-drug interactions?
Generally, no. Similar to FMOs, they are less prone to the induction and inhibition mechanisms that plague CYPs. This relative lack of interaction potential makes them potentially attractive targets for designing prodrug activation strategies.
Okay, great. We've explored phase 1, the CYPs, FMOs, EHs, and carboxylesterases. Now let's move decisively into phase 2, the conjugation reactions. These are sometimes called synthetic reactions because they involve adding a molecule and increasing the molecular mass.
Yes, they conjugate or add another molecule onto the drug or its phase 1 metabolite. And as we discussed earlier, this addition almost always terminates the biological activity of the drug, essentially making it unable to bind its target anymore.
Except for those few interesting exceptions, like morphine 6-glucuronide or minoxidil sulfate, which are actually more active than the parent drug.
Right, those are rare but important reminders that biology always has exceptions. But the main goal, the overwhelming outcome of phase 2, is generally inactivation and, crucially, producing metabolites with significantly increased water solubility.
And sulfation and glucuronidation seem particularly important for boosting that water solubility.
They are key processes for that, yes. Adding a highly polar sulfate or glucuronic acid moiety, even with only a minor change in overall charge, drastically increases the compound solubility in water. And that's essential for facilitating its transport out of tissues and its elimination from the body.
And these reactions require specific cofactors, right? The enzymes don't just pull the sulfate or glucuronic acid out of thin air.
Correct. They are cofactor dependent. The UGTs, glucuronidation, require UDP glucuronic acid, UDPGA, as the donor of glucuronic acid. The SULTs, sulfation, require 3-put-aphosphodenosine 5-site-aphososulfate, or PPS, as the sulfate donor. These cofactors provide the molecule that gets attached to the substrate.
And the substrates often already have a functional group, maybe added in phase 1. that acts as the attachment point.
Often, yes. That hydroxyl aminocarboxyl group added in phase 1 is a perfect handle for phase 2 conjugation. But it's worth remembering that some drugs or chemicals can undergo phase 2 conjugation directly without needing a preceding phase 1 step if they already have a suitable functional group. Acetaminophen is an example of a drug that directly undergoes both glucuronidation and sulfation.
Location-wise, most phase 2 enzymes are in the cytosol, but glucuronidation is different. It happens in the ER lumen.
Glucuronidation, catalyzed by UGTs, primarily occurs associated with the ER membrane, with the active site facing the lumen, the inside space, of the ER. The cofactor, UDPGA, needs to be transported into the ER lumen. Most other major phase 2 transferases, GSTs, LTs, NATs, are primarily located in the cytosol.
And generally, phase 2 reactions are faster than phase 1.
Yes, the catalytic rates of phase 2 enzymes are generally faster than those of the CYPs.
So if a drug goes through both phase 1 and then phase 2, the overall rate of elimination is often limited by that slower phase 1 step.
That's frequently the case. Phase 1 can be the bottleneck. But the speed and the dramatic increase in hydrophilicity provided by the phase 2 reactions ensure that once a compound has a suitable site for conjugation, it can be very efficiently tagged for elimination and detoxification.
Okay, let's do a deep dive into glucuronidation and the UGTs now. They seem incredibly important based on the overview.
UGTs, or uridine diphosphate glucuronyl transferases, are absolutely crucial. They catalyze the transfer of glucuronic acid from that cofactor UDPGA onto a substrate molecule. This forms a specific type of glycosidic bond, creating metabolites called glucuronides.
And these glucuronides can later be cleaved by other enzymes called glucuronidases.
Yes, that becomes important when we talk about enterohepatic recirculation.
What kinds of chemical groups on the substrate can UGTs attach glucuronic acid to? Is it specific?
They're actually quite versatile. They can target alcoholic and phenolic hydroxyl, OH groups, carboxyl, COH groups, sulfuryl groups, carbonyl groups, and even various types of amines, primary, secondary, and tertiary. This broad range of potential acceptor sites allows them to handle a vast diversity of substrates.
Hundreds of different compounds, the material says, pharmaceuticals, things we eat, environmental chemicals, and even really important endogenous substances like hormones, bile acids, retinoids, and bilirubin.
Exactly. Glucuronidation is absolutely essential for the excretion of a huge number of therapeutic agents, yes, but also for managing the levels of many key bodily compounds. Bilirubin clearance, for example, is entirely dependent on UGTs.
Is UGT expression constant, or does it vary?
Oh, it's highly regulated. Expression is coordinated, it's tissue-specific, and it can be induced, meaning levels can increase in response to certain signals or exposures.
And highest levels are where we expect, liver and GI tract.
Yes, highest levels are in the GI tract and liver. And interestingly, as we touched on, the small intestine can actually have an even higher concentration of UGTs per gram of tissue weight than the liver. This makes intestinal glucuronidation very important for the first-pass metabolism of many oral drugs.
And once the glucuronides are formed, how are they eliminated?
They're typically transported out of the cell. From liver cells, they can enter the bloodstream for excretion by the kidneys into urine, or they can be actively transported into the bile for elimination via the feces. Generally, higher molecular weight glucuronides are preferentially shunted into the bile pathway.
And this bile excretion route can lead to something called enterohepatic recirculation. Can you walk us through that?
Sure, it's a really important concept. So glucurides excreted from the liver into the bile enter the small intestine. They travel down into the large intestine. Now, bacteria residing in our large intestine produce enzymes called glucuronidases.
And these bacterial enzymes can chop the glucuronic acid back off the drug.
Precisely. They cleave that glucuronide linkage, releasing the original drug or metabolite, plus free glucuronic acid. Now, if the original molecule that's released is lipophilic enough,
It get reabsorbed.
it can be reabsorbed from the large intestine, usually via passive diffusion or maybe apical transporters, back into the portal circulation, heading back to the liver and re-entering the systemic circulation.
So the body essentially gets another exposure to the active compound, extending its time in the body.
It can, yes. This recirculation effectively delays the ultimate excretion of the compound and can significantly extend its apparent half-life in the body. It's a natural and important process for conserving things like bile acids, but it significantly affects the pharmacokinetics of certain drugs too, including that irinotecan metabolite SN38.
How many human UGT genes are there? Is it a big family?
We have 19 functional UGT genes identified in humans. They're organized into two main families. 9 genes are located together in a cluster called the UGT1A locus on chromosome 2. The other 10 genes belong to the UGT2 family, primarily located on chromosome 4.
Are all 19 heavily involved in drug metabolism?
The ones most consistently implicated in metabolizing drugs are generally UGT1A1, 1A3, 1A4, 1A6, 1A9, and UGT2B7. The UGT2 family seems perhaps more specialized towards endogenous compounds, although there's overlap.
The structure of that UGT1A locus on chromosome 2 is described as quite unique. How does that work?
It is fascinating genetically. It's a large region, about 200 kilobases long. It has a unique structure where there's a tandem array of different cassette exon 1s lined up at the beginning. Each of these different exon 1s encodes the unique N-terminal part, about 280 amino acids, of a different UGT1A protein.
Okay.
Then downstream from all these unique exon 1s, there were 4 common 3' exons. These common exons encode the C-terminal part of the protein, about 245 amino acids, which is shared by all the UGT1A proteins generated from this locus.
So each specific UGT1A enzyme is made by splicing one unique exon 1 together with those 4 common exons.
Exactly. It's like plugging different unique cartridges, exon 1s, into a common player, the common exons. And it's the divergence, the differences in the sequences of those exon 1s, that gives each UGT1A enzyme its distinct substrate specificity, determining which drugs or compounds it prefers to glucuronidate.
That structure also has implications for mutations, right?
It does. A mutation in one of the common exons would affect all the UGT1A enzymes produced from that locus. But a mutation within a specific exon 1 would only affect that single UGT1A protein.
And there are many known variations in these exon 1 regions, over 100.
Yes, over 100 allelic variants have been identified just within the various exon 1 sequences of the UGT1A locus, and many of these lead to reduced enzyme activity. This genetic variability is a major reason why individuals differ so much in their capacity to glucuronidate certain substrates.
And UGT1A1 in particular seems clinically vital, like essential for life.
It truly is. UGT1A1 is the primary and arguably the only physiologically relevant enzyme responsible for glucuronidating bilirubin. And this step, bilirubin glucuronidation by UGT1A1, is the rate-limiting factor in clearing bilirubin from the body.
Remind us where bilirubin comes from.
Bilirubin is the toxic breakdown product of heme, which comes mostly from the turnover of old red blood cells hemoglobin. It's naturally hydrophobic and travels in the blood bound to albumin. It absolutely must be glucuronidated by UGT1A1 to make it water-soluble so it can be excreted, primarily into bile.
And if this process fails...
If bilirubin glucuronidation fails or is severely impaired, unconjugated bilirubin builds up in the blood. That's called unconjugated hyperbilirubinemia, and clinically it manifests as jaundice, the yellowing of the skin and eyes. The delayed expression and lower activity of UGT1A1 in newborns is the reason why neonatal jaundice is so common.
And there are inherited conditions caused by severe UGT1A1 deficiency.
Yes, several. There are over 40 known genetic lesions within the UGT1A1 gene that cause different inherited forms of unconjugated hyperbilirubinemia. The most severe is Crigler-Najjar syndrome type 1.
What happens in type 1?
It results from mutations, either in exon 1 or the common exons, that cause a complete or near complete lack of bilirubin glucuronidation activity. Unconjugated bilirubin accumulates to extremely high toxic levels, which can cause severe brain damage, kernicterus. Infants with CN1 require intensive phototherapy. Blue light helps break down bilirubin in the skin, and typically need a liver transplant eventually to survive.
Type 2 is less severe.
Crigler-Najjar syndrome type 2 is also serious, but less severe than type 1. It usually involves mutations, often in the promoter region or causing reading frame shifts, that greatly reduce UGT1A1 expression and activity, but don't completely eliminate it. There's still significant hyperbilirubinemia and a risk of neurological toxicity, but these patients often show some response to treatment with drugs like phenobarbital, which can induce the expression of the remaining functional UGT1A1 enzyme.
And then there's the much more common one, Gilbert syndrome.
Right. Gilbert syndrome is considered a benign condition. It affects a surprisingly large percentage of the population. Estimates range from maybe 8% up to 23%, depending on the population studied. It's characterized by mildly elevated levels of unconjugated bilirubin, typically fluctuating but maybe 100% to 300% higher than normal, often noticed during illness or stress.
And what causes Gilbert syndrome?
The most frequent cause is a very common polymorphism in the promoter region of the UGT1A1 gene. Specifically, it's in a variation in the number of TA repeats in a region called the TATA box. The normal allele, wild type, has 6 TA repeats, ATA, 6 TAA. The common variant allele associated with Gilbert's, often called UGT1A128, has 7 TA repeats, ATA 7 TAA.
And that extra TA repeat just slightly reduces how much UGT1A1 enzyme is made.
Exactly. It leads to moderately reduced hepatic expression of the UGT1A1 protein, resulting in slightly lower bilirubin glucuronidation capacity, and thus the mild hyperbilirubinemia.
It's generally considered harmless, right? Maybe even protective against cardiovascular disease because bilirubin has antioxidant properties.
It's generally considered benign, yes, and the potential protective effect is an area of ongoing research. However,
There's a catch related to drugs.
there is. Having this reduced UGT1A1 capacity, even if mild, can make individuals with Gilbert syndrome more prone to adverse drug reactions from medications that are primarily substrates for UGT1A1 elimination.
How does that work? Competition.
Essentially, yes. If a drug relies heavily on UGT1A1 for its glucuronidation and elimination, individuals with Gilbert syndrome have less enzyme activity to handle both the drug and their baseline bilirubin load. The drug competes with bilirubin for that limited enzyme capacity.
So what happens?
This can lead to 2 potential problems: a more pronounced increase in bilirubin levels, drug-induced jaundice, and perhaps more importantly, reduced clearance, and thus elevated plasma levels of the drug itself, increasing the risk of drug toxicity.
Like with the cancer drug irinotecan again, that seems to be a recurring example.
It's a prime example because the consequences can be severe. As we said, irinotecan is activated to SN38. SN38 is then inactivated and prepared for elimination primarily by UGT1A1 glucuronidation, mainly into bile. But don't forget the intrahepatic recirculation.
Right, where intestinal bacteria cleaves the glucuronide, releasing SN38 back into the gut where it can be reabsorbed.
Exactly. So in patients with Gilbert syndrome who have insufficient UGT1A1 activity to efficiently glucuronidate the SN38 that's circulating and being reabsorbed.
SN38 levels can build up significantly higher than in people with normal UGT1A1 activity.
Correct. And this accumulation is strongly associated with severe toxicities, particularly hematological ones like profound leukopenia and neutropenia, low white blood cell counts, and also severe gastrointestinal toxicity, like debilitating diarrhea and inflammation of the bowel, ileocolitis, likely due to high concentrations of SN38 accumulating in the intestinal lining.
So knowing a patient's UGT1A1 status, like having the 28 allele, is really important before giving irinotecan.
It's become increasingly recognized as crucial for predicting toxicity risk and potentially guiding dosing decisions, especially since cancer drugs often have a narrow therapeutic window.
The HIV drug atazanavir also comes up here in relation to Gilbert's. Is it a substrate too?
Atazanavir is interesting because it's not primarily a UGT1A1 substrate itself, but it acts as an inhibitor of UGT1A1 activity.
Ah, so it blocks the enzyme.
Yes. Therefore, it can cause elevated bilirubin levels, hyperbilirubinemia, in pretty much any patient taking it. But this effect is much more pronounced, sometimes severely so, in patients who also have Gilbert syndrome, because their baseline UGT1A1 capacity is already reduced.
So it's interaction between a drug inhibiting the enzyme and an underlying genetic predisposition for lower enzyme levels.
Exactly. And severe hyperbilirubinemia can also occur if patients have other inactivating mutations in related UGT genes, like UGT1A3 or 1A7. This interplay between drug-induced inhibition and underlying genetic mutations in metabolic pathways is a really significant concern in clinical practice.
That was a really comprehensive look at UGTs and their clinical importance. What about the next phase 2 pathway? Sulfation, carried out by SULTs.
Right, SULTs or sulfotransferases. These are generally cytosolic enzymes, meaning they're found in the main fluid compartment of the cell. They catalyze the transfer of a sulfate group, SO3, from that high-energy cofactor PPS onto hydroxyl, OH, or sometimes amine NH2 groups of their substrates.
And they handle both endogenous compounds and xenobiotics.
Yes, they play roles in metabolizing both. Humans have about 13 identified SULT isoforms, which are grouped into 4 families, SULTs T1, 2, 4, and 6. Now, like other enzyme families, there are major differences in SULTs between species, which again makes extrapolating human sulfation patterns from animal studies unreliable.
Do SULTs play important roles in normal body function beyond just getting rid of foreign chemicals?
Oh, absolutely. They are crucial for homeostasis. For example, one isoform, ULTA-T2B1B, is important in the skin for cholesterol sulfation and regulating skin cell differentiation. SULT-T2A1 is key in the fetal adrenal gland for sulfating DHEA, a precursor needed for estrogen production during pregnancy. SULT-T1A3 and 1A4 are involved in sulfating catecholamine neurotransmitters. SULT-T1E1 sulfates estrogens. Many important endogenous compounds actually circulate in our bloodstream predominantly as sulfate conjugates.
Do individual SULTs have broad substrate specificity like some CYPs, or are they more specific?
It varies. Some isoforms seem to have unique specific endogenous substrates. Others, particularly within the SULT-T1 family, which is majorly involved in xenobiotic metabolism, can be quite promiscuous.
Which SULT is most important for drugs?
SULT-1A1 is considered quantitatively and qualitatively the most important salt in the human liver for xenobiotic metabolism. It's often referred to as a phenol salt because it can sulfate a broad range of phenolic compounds molecules with an OH group attached to an aromatic ring. This includes common drugs like acetaminophen and minoxidil, as well as the synthetic estrogen 17-ethinyl estradiol found in oral contraceptives.
Is sulfation always a detoxification step? Does it make things less harmful?
Mostly, yes. Just like glucuronidation, adding the highly polar sulfate group greatly increases water solubility and facilitates excretion,
Right.
so it's primarily a detoxification pathway. However, there's a potential dark side.
Oh.
In certain cases, particularly with some types of environmental or dietary procarcinogens, the addition of a sulfate group can actually lead to the formation of a reactive metabolite. The sulfate group is electron withdrawing, and if it leaves the molecule, heterolytic cleavage, it can generate a highly reactive electrophilication that can damage DNA. This mechanism has been documented with certain carcinogens found in things like burnt meat or environmental pollutants. It's less common than detoxification, but it's a known mechanism of metabolic activation.
So understanding variations in SULT activity, like polymorphisms, could potentially link individual genetic differences to susceptibility to cancers caused by these environmental exposures.
Exactly. That's an active area of research. There's particular interest in potential links between common polymorphisms in SULT-1 and cancer risk, although the evidence is still evolving. We know that genetic variations, including copy number polymorphisms, having extra or fewer copies of the gene, exist for SULT-1A1, 1A3, and 1A4, and these contribute significantly to the inter-individual variation we see in sulfation capacity. Understanding the structure, activity, regulation, and genetics of SULTs is key to unraveling these complex links between metabolism, environment, and diseases like cancer, as well as their roles in reproduction and development.
Yeah. Another major phase 2 process we need to cover is glutathione conjugation, mediated by the GSTs.
Right, GSTs or glutathionase transferases. These are absolutely critical cellular defense enzymes. Their main job is to catalyze the transfer of glutathione, which is a tripeptide molecule, onto reactive electrophiles.
What's glutathione made of again?
It's glutamic acid, cysteine, and glycine linked together.
And GSTs attach this glutathione molecule to reactive electrophiles.
Yes. The reaction typically forms a thioether linkage between the sulfur atom of the cysteine residue in glutathione and an electrophilic atom on the substrate. This conjugation effectively neutralizes the reactive electrophile, preventing it from damaging cellular macromolecules like DNA, RNA, and proteins. It's a major protective mechanism.
Glutathione itself is important for maintaining the cell's redox state, right? Keeping things reduced.
It is. The ratio of reduced glutathione, GSH, to its oxidized form, GSSG, is a critical indicator of the cell's redox balance. Severe depletion of cellular TSH leaves cells vulnerable to oxidative damage, which is linked to numerous health problems.
What kind of substrates do GSTs typically target?
Their substrates are typically electrophilic, often containing heteroatoms like oxygen, nitrogen, or sulfur, and they're usually hydrophobic enough to associate with cellular components.
Does this reaction always need the GST enzyme?
Well, the concentration of glutathione itself inside cells, especially liver cells, is very high, around 10 millimolar. So some non-enzymatic reaction between glutathione and very strong electrophiles can occur spontaneously. However, GST enzymes are also incredibly abundant. They can make up something like 10% of the total soluble protein in a liver cell. This high enzyme concentration ensures that the conjugation reaction is highly efficient for a wide range of electrophiles.
The material mentions cytosolic GSTs also acting as binding proteins, ligandins.
Yes, some cytosolic GSTs, historically called ligandins, have high affinity binding sites for various compounds, including steroids, bile acids, bilirubin, hormones, and certain environmental toxicants, even without necessarily catalyzing a reaction. They can help sequester these compounds or facilitate their transport.
Are there many different GST enzymes?
Yes, there are over 20 known human GSTs. They are broadly classified into cytosolic and microsomal membrane-associated forms. The cytosolic GSTs are generally considered more important for drug and xenobiotic metabolism, and are further divided into several classes based on structure and sequence: alpha, mu, omega, pi, sigma, theta, zeta. Some can even form heterodimers between different subunits. The microsomal GSTs, MAPIG superfamily, seem more involved in endogenous pathways, like the metabolism of leukotrienes and prostaglandins.
The material talks about overcapacity in this system, meaning the high levels of both glutathione and GSTs provide a significant buffer against damage.
That seems to be the case, yes. Under normal circumstances, the high concentrations of both co-substrate, GSH, and the enzymes, GSTs, mean that very few reactive electrophilic molecules manage to escape conjugation and cause damage. The concern arises when the system is overwhelmed or compromised.
How could it be compromised?
Well, cellular GSH levels can be depleted, for instance, by exposure to very high doses of certain drugs that consume GSH during their metabolism. Or the GST enzymes themselves might be dysfunctional due to genetic polymorphisms.
Like the acetaminophen overdose example.
That's the classic illustration. Normally, acetaminophen is safely metabolized primarily via phase 2 glucuronidation and sulfation. However, a small portion is oxidized by CYPs, mainly CYP2E1 and CYP3-4, to a highly reactive toxic electrophilic metabolite called NAPQI.
And normally, NAPQI is detoxified
by conjugation with glutathione, catalyzed by GSTs. But in an overdose situation, the glucuronidation and sulfation pathways become saturated. More acetaminophen gets shunted down the CYP pathway, producing large amounts of NAPQI. This rapidly depletes the liver stores of glutathione.
And once the glutathione runs out,
NAPQI has nothing left to react with except cellular macromolecules, particularly proteins in the liver cells. This covalent binding leads to cellular dysfunction, oxidative stress, and eventually cell death, causing the severe hepatic necrosis characteristic of acetaminophen toxicity.
And the antidote, N-acetylcysteine, works by replenishing glutathione.
Exactly. N-acetylcysteine provides cysteine, which is the rate-limiting precursor for glutathione synthesis, helping the liver replenish its GSH store so it can resume detoxifying NAPQI.
You mentioned GST polymorphisms. Are those common?
Yes. Virtually all GST genes are known to be polymorphic. Two particularly well-studied examples involve the mu class, GSTM1, and the theta class, GSTT1. For both of these genes, there are common null alleles, or gene deletions, often designated GSTM10 and GSTT10.
A null phenotype means individuals who inherit two copies of this allele completely lack functional expression of that specific GST enzyme.
Correct. Individuals who are homozygous for these null alleles, GSTM1 null or GSTT1 null, have essentially zero activity of that particular GST enzyme.
And does this put them at higher risk?
It can, yes. They may be predisposed to toxicity from specific xenobiotics or drugs that are selectively detoxified by the missing enzyme. For instance, the GSTM10 null allele is quite common, present in about 50% of Caucasians, and has been epidemiologically linked, albeit sometimes controversially, to an increased risk of certain cancers, like lung, colon, and bladder cancer, potentially due to impaired detoxification of environmental carcinogens.
And GSTT1?
The GSTT10 null allele is also common, particularly in some Asian populations, up to 60% in Chinese and Koreans. It has been linked to adverse effects and increased toxicity with certain cytostatic cell-killing cancer drugs, presumably because those drugs or their metabolites aren't being cleared effectively by GSTT1 conjugation. So yes, GST polymorphisms can definitely influence both drug efficacy and the severity of side effects.
Interestingly, the material also discusses GSTs in the context of cancer cells developing resistance to chemotherapy.
Yes, that's another important aspect. It's frequently observed that many types of cancer cells show elevated levels or overexpression of certain GST isoforms.
And this helps them resist the chemo. How?
It's thought to contribute to resistance in a couple of ways. First, if the chemotherapy drug itself, or one of its active metabolites, is a substrate for the overexpressed GST, the cancer cell can simply detoxify and eliminate the drug more efficiently. But resistance can also occur even with drugs that aren't direct GST substrates.
How does that work?
Overexpression of certain GSTs, particularly the pi class, GSTP1, seems to interact with cellular signaling pathways, like certain MAP kinase pathways, J and K, P38. By reducing the activity of these stress-activated pathways, the GST overexpression can make cancer cells more resistant to undergoing apoptosis, or programmed cell death, which is what many chemotherapy drugs are designed to trigger.
So this has led to therapeutic strategies trying to inhibit GSTs in cancer treatment, to resensitize the tumors.
Exactly. The idea is that if you can inhibit the overexpressed GSTs, you might make the cancer cells more vulnerable to standard chemotherapy agents. The material mentions a prodrug called TLK199.
How does that work?
TLK199 is designed to be converted by other cellular enzymes, esters, into an active GST inhibitors called TLK117. Studies have shown that TLK117 can potentiate or enhance the anticancer effects of other agents when given in combination.
And the opposite strategy, exploiting the elevated GST levels often found in cancer cells to actually activate a poor drug.
That's another clever targeted approach. A different prodrug called TLK286 was designed specifically to be a substrate for GSTs, particularly those overexpressed in tumors.
So the GST enzyme itself activates the killer drug.
Yes. When TLK286 encounters high levels of GST in a cancer cell, the enzyme catalyzes a reaction, elimination, that releases two things: a glutathione conjugate and an active nitrogen mustard. Nitrogen mustards are potent alkylating agents that damage DNA and kill the cell. So it's turning the tumor's own defense mechanism, high GST, into the trigger for its destruction.
That's a fascinating targeted activation strategy. Okay, next up for phase 2, N-acetylation, carried out by gnats.
Right, gnats, or N-acetyltransferases, these are cytosolic enzymes. Their primary role is to metabolize drugs and environmental chemicals that contain either an aromatic amine group, an NH2 attached to a benzene-like ring, or a hydrazine group, NHNH2.
And they do this by adding an acetyl group.
Yes, they transfer an acetyl group, CH3CO, from the cofactor acetyl-CoA onto that MN or hydrazine group on the substrate.
Now, this is one of the phase 2 reactions that might actually make the metabolite less water-soluble, unlike glucuronidation or sulfation.
It often does, yes. Adding the neutral acetyl group usually neutralizes the positive charge that the amine group might have at physiological pH. This loss of charge can decrease water solubility compared to the parent amine.
And gnats are known for being highly polymorphic.
Extremely so. They are among the most polymorphic of all human xenobiotic metabolizing enzymes.
This is where the whole concept of acetylator phenotype comes from in pharmacogenetics, isn't it?
Exactly. This is actually one of the very first examples of pharmacogenetics discovered decades ago. It was observed with the tuberculosis drug isoniazid, INH. Doctors noticed that some patients treated with standard doses developed toxic side effects, like peripheral neuritis, nerve damage, or CNS effects.
And they found a difference in metabolism.
Yes. When they measured the drug and its metabolites in urine, they found that the patients experiencing toxicity excreted much more of the unchanged isoniazid drug and much less of its primary metabolite, acetylisoniazid. This led to the classification of people into rapid acetylators and slow acetylators.
And the slow acetylator phenotype was linked to the toxicity.
Yes, the slow acetylator phenotype clearly predisposed individuals to developing INH toxicity at standard doses, and it was soon established that this phenotype was genetically determined.
How many human NAT genes are there?
Humans have two functional NAT genes, NAT1 and NAT2, and there are many known genetic variants, or alleles, for both genes, over 25 alleles described for NAT2 alone.
And the slow acetylation pattern is usually due to...
The slow acetylator phenotype typically results from inheriting two copies, being homozygous, of specific variant alleles of the NAT2 gene that lead to reduced enzyme activity or stability. The NAT2 polymorphism is what primarily accounts for the classic rapid versus slow acetylator patterns observed with drugs like isoniazid.
Are there many common drugs metabolized by gnats besides isoniazid?
Yes, a significant number. The list includes drugs like acebutolol, aminoglutathione, aminosalicylic acid, amrinone, benzocaine, caffeine, partially, clonazepam, dapsone, hydrolazine, nitrazepam, phenelzine, procainamide, and importantly, the whole class of sulfonamide antibiotics.
So for slow acetylators taking these drugs, the toxicity can basically look like an overdose at normal doses.
It often does because the drug isn't being cleared as efficiently. Therefore, knowing a patient's acetylator phenotype, which can be predicted by genotyping for NAT2 variants, is really important for optimizing therapy with drugs that have aromatic amine or hydrazine groups. Dose reduction or increasing the dosing interval might be recommended for slow acetylators to prevent toxicity and ensure positive outcomes.
Any other examples of toxicity?
With the antihypertensive drug hydrolyzine, for instance, slow acetylators given standard doses can experience potentially dangerous side effects like extreme hypotension, low blood pressure, and tachycardia, rapid heart rate.
And those hypersensitivity reactions sometimes seen with sulfonamide antibiotics are also linked to slow acetylation.
Yes, slow acetylators appear to be more susceptible to these reactions. It's thought that sulfonamides can be metabolized via an alternative pathway, an oxidation by CYPs, to form reactive hydroxylamines. These can then react with cellular proteins, forming haptens that trigger an immune response, leading to hypersensitivity. Slow acetylators accumulate more parent drug, potentially pushing more down this alternative pathway.
The material also discusses tissue-specific NAT activity and how that plays into toxicity, particularly with environmental carcinogens.
Right. It's not just about overall acetylation status. NAT1 is expressed quite widely in many tissues throughout the body, whereas NAT2 expression is predominantly found in the liver and the lining of the GI tract.
And both NAT1 and NAT2 can be involved in activating certain carcinogens.
Yes, particularly bicyclic aromatic amines, which are found in things like tobacco smoke and cooked meats. While direct N-acetylation of these compounds, adding the acetyl group directly to the amine, is generally a detoxification step, leading to stable, excretable metabolites.
There's another pathway.
Yes. These aromatic amines can first undergo N-hydroxylation, adding an OH group to the nitrogen, by CYP enzymes. Then either NAT1 or NAT2 can catalyze O-acetylation, adding the acetyl group to that oxygen. This N-O-acetylated metabolite is unstable. It can non-enzymatically release the acetyl group, leaving behind a highly reactive nitrenium ion, which is a potent mutagen capable of binding to DNA.
So acetylation can be detoxification or activation, depending on the specific reaction and substrate.
Exactly. And the tissue location matters. Consider someone exposed to these bicyclic aromatic amines.
Yes.
If they are a fast N-2 acetylator, they will efficiently detoxify these amines via direct N-acetylation in the liver. But if they are a slow NAT2 acetylator, they will accumulate the parent amines. These accumulated amines are then more likely to undergo N-oxidation by CYPs, forming the N-hydroxy metabolites.
And these N-hydroxy metabolites are excreted in urine.
Yes. But when they reach the bladder epithelium, the lining of the bladder, where NAT1 activity is high, even if NAT2 is low.
NAT1 can perform that problematic O-acetylation step there.
Right. NAT1 in the bladder can O-acetylate the N-hydroxy metabolite, leading to the local generation of that mutagenic nitrenium ion right there in the bladder tissue.
So epidemiological studies have linked slow NAT2 acetylators exposed to these compounds to an increased risk of bladder cancer.
Yes, numerous studies support this link. It's a complex pathway. A deficiency in one enzyme, NAT2, shunts metabolism towards another pathway, CYP N-oxidation, generating a precursor metabolite that is then activated by a different enzyme, NAT, in a specific target tissue, bladder, leading to increased cancer risk. It beautifully illustrates the interplay between genetics, different metabolic pathways, and tissue-specific enzyme expression in determining toxicity.
Absolutely fascinating complexity. Finally, for phase 2, let's talk about methylation carried out by MTs.
Right, methyltransferases or MTs. These enzymes catalyze the addition of a methyl group, known as CH3, onto oxygen, O-methylation, nitrogen N-methylation, or sulfur S-methylation atoms in their substrates.
Are there many different MTs?
Humans express several types, including catechol O-methyltransferase, COMT, which has 2 isoforms, various N-methyltransferases, at least 3 types like NNMT, PNMT, HNMT, phenylethylamine N-methyltransferase, PNMT, histamine N-methyltransferase, HNMT, thiopurine S-methyltransferase, TPMT, and thiolmethyltransferase, TMT.
And they use a specific methyl donor.
Yes, they all use S-adenosylmethionine, SAM, or Adamet, as the donor of the methyl group. They generally function as monomers, single protein units, and despite catalyzing a similar reaction, don't share extensive sequence similarity except for a signature sequence involved in SAM binding. They tend to have relatively high substrate specificity compared to, say, CYPs or UGTs.
And they metabolize important endogenous compounds too, not just drugs.
Definitely. For example, PNMT is responsible for methylating norepinephrine to produce epinephrine, adrenaline. HNMT methylates histamine. CMT is famous for methylating catechol neurotransmitters like dopamine and norepinephrine, but it also methylates drugs containing a catechol structure like methyl-dopa or even MDMA, ecstasy. NNMT can methylate serotonin, tryptophan, and various pyridine compounds like nicotinamide and nicotine.
And TPMT is particularly important clinically thiopurine S-methyltransferase.
Absolutely critical. TPMT catalyzes the S-methylation, adding a methyl group to a sulfur atom of a class of drugs called thiopurines. This includes azathioprine, AZA, 6-mercaptopurine, 6-MP, and thioguanine.
What are these drugs used for?
They are widely used as immunosuppressants in conditions like inflammatory bowel disease, Crohn's, ulcerative colitis, and autoimmune disorders, and also as anti-cancer agents, particularly in the treatment of childhood acute lymphoblastic leukemia, ALL.
And TPMT's role is to detoxify these drugs.
Essentially, yes. 6-MP, for instance, is actually a prodrug. It needs to be converted by other enzymes, part of the purine salvage pathway involving HTPRT, into active metabolites called 6-thioguanine nucleotides, 6-TGNs. These 6-TGNs are the cytotoxic agents. They get incorporated into DNA and RNA and disrupt cellular processes, which is how they kill cancer cells or suppress immune cells.
So where does TPMT fit in?
TPMT provides an alternative metabolic route. It inactivates 6-MP directly by methylating its sulfur atom. This methylated metabolite cannot be converted into the active toxic 6-TGNs. So TPMT essentially diverts the drug away from the activation pathway towards an inactive elimination pathway.
So if someone has a genetic deficiency in TPMT activity, which happens, right?
Yes, it does. Due to common genetic polymorphisms in the TPMT gene, about 1 in 300 individuals inherit 2 non-functional alleles and have virtually no TPMT activity. About 10% of people are heterozygous and have intermediate activity.
So in those individuals with little or no TPMT activity,
if they're given standard doses of thiopurine drugs, they cannot effectively detoxify the drugs via methylation. This leads to a massive shunting of the drug down the activation pathway, resulting in the accumulation of extremely high toxic levels of the active 6-TGNs.
And the consequence?
Severe, potentially life-threatening myelosuppression, a drastic drop in the production of white blood cells, red blood cells, and platelets by the bone marrow.
So testing for TPMT status is crucial before starting these drugs.
Absolutely. Identifying individuals with deficient TPMT activity, either by genotyping for the common non-functional alleles, or by measuring TPMT enzyme activity in red blood cells, is now considered standard practice before initiating thiopurine therapy. For patients with low or absent activity, the drug dose needs to be drastically reduced, or an alternative therapy chosen altogether. This is a truly life-saving application of pharmacogenetics.
That's an incredibly powerful example of how understanding of the genetics of just one metabolic enzyme can directly impact patient safety and guide personalized dosing decisions.
It really is a cornerstone example of pharmacogenetics in routine clinical action.
Okay. We've covered the phases, the major enzymes, their mechanisms, locations, substrate specificities, polymorphisms. Let's try to tie this all together now. What's the overall role of all this complex xenobiotic metabolism in ensuring that drugs are used safely and effectively?
Well, at its most fundamental level, metabolism coupled with excretion is our body's way of protecting itself. It prevents foreign substances, including drugs, from accumulating indefinitely to potentially toxic levels. For drugs specifically, metabolism typically serves to inactivate them and, crucially, make them more water-soluble to facilitate their removal from the body. This process ultimately controls how long a drug stays active in the body, determining its biological half-life.
And the speed and extent of this metabolism directly dictates both how effective a drug will be and how likely it is to cause toxicity.
That's absolutely right. It's a balancing act. Adverse drug reactions, ADRs, are a massive problem in healthcare. The statistics are pretty sobering. They cost healthcare systems billions of dollars annually and contribute to, estimates suggest, over 100,000 deaths each year just in the U.S. And it's estimated that something like 56%, over half, of drugs associated with ADRs are substrates for these metabolic enzymes.
So if a drug is metabolized too quickly, what happens?
It gets cleared from the body too fast. Its plasma concentrations might not reach, or might not stay within, the therapeutic window needed for efficacy. You effectively lose the drug's benefit. This can happen if an individual naturally has very high enzyme activity, an ultra-rapid metabolizer phenotype due to genetics, or if the relevant enzyme activity has been induced by other factors like co-administered drugs or supplements.
And the flip side, if metabolism is too slow,
then the drug isn't cleared efficiently. It accumulates in the body, leading to higher than intended plasma concentrations, an increased AUC, area under the plasma concentration time curve, and a significantly increased risk of dose-dependent toxicity. This is what happens when enzyme activity is inhibited.
And inhibition can come from various sources.
Yes, commonly from other drugs competing for the same enzyme, or from direct enzyme inhibitors found in the diet, like the classic grapefruit juice example, inhibiting intestinal CYP3A4, or potentially from underlying disease states affecting enzyme function.
And genetic predisposition, those polymorphisms we discussed, are a huge part of this variability in both efficacy and toxicity, linking back strongly to pharmacogenetics.
Yes, absolutely. Heritable differences in metabolic enzyme function due to common genetic polymorphisms in genes like the CYPs, especially 2D6, 2C19, 2C9, and the UGTs, like UGT1A1, significantly influence how individuals will metabolize a vast number of drugs.
Gilbert syndrome, with its reduced UGT1A1 capacity, is a prime example again.
It is. It directly leads to reduced glucuronidation of drugs that are UGT1A1 substrates, like SN38 from irinotecan. This increases their AUC, prolongs their exposure, and significantly increases the risk of severe toxicity, which is especially problematic for drugs like cancer chemotherapies that already have a narrow therapeutic index.
It seems like these metabolic enzymes are involved in adverse reactions across nearly every class of drug imaginable.
They are absolutely central players in determining drug disposition and response. And the mechanisms of induction and inhibition, particularly involving the CYP and UGT enzymes, are extremely common causes of drug interactions in ADRs.
This is why, in drug development, understanding metabolism is non-negotiable.
It's absolutely essential. For any new drug application, NDA, submitted to regulatory agencies like the FDA, the sponsor must provide a thorough characterization of the drug's metabolism. They need to identify the primary enzymes involved, map out the major metabolic pathways and metabolites, and assess the potential toxicity of those metabolites.
And there's even a trend now to try and design drugs to avoid certain pathways.
Yes, there's a growing trend in medicinal chemistry to try and design new small molecule drugs where possible to avoid heavy reliance on metabolism via the major oxidative CYP pathways, like CYP3A4, UD6, 2C9, 2C19. The goal is to minimize the potential issues arising from the known genetic variability and susceptibility to drug interactions associated with these enzymes.
You mentioned induction earlier, how drugs can increase enzyme levels by activating nuclear receptors. Can you explain that transcriptional activation mechanism in a bit more detail? How does a drug binding a receptor lead to more enzyme?
Sure. So certain xenobiotics and drugs can act as ligands molecules that bind specifically to certain proteins inside the cell called nuclear receptors. Many of these receptors belong to the steroid hormone receptor superfamily and function as transcription factors.
Meaning they control gene expression.
Exactly. When the ligand, the drug or xenobiotic, binds to its specific nuclear receptor, the receptor often undergoes a conformational change. It then typically partners up with another nuclear receptor, most commonly the retinoid X receptor, or RX star, forming a heterodimer complex.
Okay, so the drug binds, the receptor pairs up, then what?
This activated receptor complex then moves into the cell nucleus, if it wasn't already there, and binds to specific short DNA sequences called response elements, located in the regulatory regions, promoters or enhancers, upstream of its target genes.
And those target genes include the genes for metabolic enzymes.
Yes. Critically, they include the genes encoding various CYPs, UGTs, SLTs, GSSTs, and also drug transporter proteins. So the receptor complex binds to the DNA near these genes. This binding event then serves as a platform to recruit other proteins called coactivators.
And coactivators help turn the gene on.
Precisely. The coactivators help to modify the chromatin structure around the gene and recruit the basic transcription machinery, like RNA polymerase II, ultimately leading to an increased rate of transcription of that target gene into messenger RNA, mRNA. More mRNA means more protein the enzyme or transporter is synthesized.
So drug binds receptor, receptor binds DNA, transcription increases, more enzyme is made.
That's the basic mechanism of nuclear receptor-mediated induction. And this can cause either autoinduction, the drug inducing its own metabolism, or induction of the metabolism of other drugs or xenobiotics that are substrates for the induced enzyme or transporter.
Are there specific receptors known to be key players in inducing drug metabolism?
Yes, several are particularly important. The material highlights the aryl hydrocarbon receptor, AHR, the constitutive androstenedione receptor, CAR, the pregnane X receptor, PXR, farnesoid X receptor, FXR, vitamin D receptor, VDR, and the peroxisome proliferator activated receptors, PPARs, along with RR and RXR, which often act as partners. Each of these receptors tends to be activated by different sets of ligands, drugs, xenobiotics, or endogenous molecules.
Let's look at a couple of those key ones. The aryl hydrocarbon receptor, AHR, what does it do?
AHR is a fascinating receptor, part of a different superfamily of transcription factors, BHLH-PAS. It has known roles in normal development, particularly CNS development, and stress responses. However, it's famously activated by certain environmental pollutants, like dioxins like AGTCDD, polycyclic aromatic hydrocarbons, PHs found in smoke and charred food, and some natural compounds.
And when AHR is activated, what genes does it induce?
Critically, it induces the expression of CYP1A1, CYP1A2, and CYP1B1.
And those are the CYPs we mentioned earlier that are involved in activating chemical carcinogens.
Yes, exactly. Many environmental and dietary procarcinogens are converted into their ultimate reactive DNA-damaging forms only after metabolism by these specific CYPs. So activation of AHR by pollutants or dietary components can potentially increase toxicity and contribute to cancer risk by ramping up the production of these activating enzymes.
Does AHR activation affect therapeutic drugs too?
It can. Even a common drug like omeprazole, a proton pump inhibitor, is known to be an AHR ligand and can induce CYP1A1 and CYP1A2. This could potentially lead to interactions if omeprazole is taken concurrently with other drugs that are substrates for these CYPs, or theoretically increase the activation of certain toxins.
Okay. Then there are the type 2 nuclear receptors, part of that steroid hormone receptor superfamily. Some were initially called orphan receptors.
Right, because when they were first discovered, based on their sequence similarity to known hormone receptors, their natural endogenous ligands weren't immediately identified. Now we know that many of these, while having endogenous roles, are also promiscuously activated by a wide range of xenobiotics and drugs. PXR, CIR, and PPRs are particularly important regulators of drug metabolism enzymes.
Let's take PXR first, the pregnein X receptor. It's activated by a synthetic steroid, pregnenolone 16-carbonitrile in rodents, but also by an incredibly diverse array of drugs in humans.
An astonishing range, yes. The list of known human PXR activators includes antibiotics like rifampicin, petrolaandomycin, calcium channel blockers like nifedipine, statins like mevastatin, older antidiabetic drugs like triglycerin, HIV protease inhibitors like ritonavir, the cancer drug paclitaxel, hyperforin from St. John's wort. It's remarkably promiscuous in what it binds.
And PXR activation has been linked to clinically significant problems like liver toxicity.
Yes, there's a significant example related to HIV treatment mentioned. Liver toxicity was sometimes observed in patients treated with combinations that included potent PXR activators, like the antibiotic rifampicin or the antiretroviral efavirenz, especially when followed by regimens containing ritonavir, another HIV protease inhibitor.
How does PXR activation cause that?
The hypothesis is that rifampicin and efavirenz strongly activate PXR. PXR activation then leads to a massive induction, very high levels, of CYP3A4 enzyme in the liver. This highly induced CYP3A4 then greatly increases the metabolism of ritonavir. While metabolism is usually detoxification, in this case, it seems to generate reactive metabolites of ritonavir. These reactive metabolites cause stress in the endoplasmic reticulum, ER stress, and ultimately damage the liver cells, hepatocytes.
So again, it's the induction leading to increased formation of a toxic metabolite, not the drug itself being directly toxic at normal levels.
Exactly. It's an indirect toxicity mediated by enzyme induction, increasing the production of a harmful intermediate. This highlights the need for strategies to potentially decrease PXR activation or mitigate the downstream pathways causing damage for safer use of drugs like ritonavir in certain combination therapies.
And St. John's wort comes up again with PXR activation.
Yes, hyperforin, a major active component of St. John's wort, is a potent activator of human PXR. As we discussed, this induces CYP3A4, leading to the increased metabolism and reduced plasma levels of many oral drugs, including the infamous interaction causing oral contraceptive failure. PXR activation also induces various drug transporters and some phase 2 enzymes, like certain SULTs and UGTs, generally contributing to a coordinated response to facilitate overall xenobiotic elimination, but sometimes with these undesirable consequences.
The species differences in PXR ligands are also highlighted as a major issue for drug development.
They are quite marked and problematic. Rifampicin, a strong human PXR activator, only activates mouse and rat PXR at much higher doses. Conversely, pregnenolone 16-carbonitrile, PCN, which is a classic potent activator of mouse and rat PXR, has minimal effect on human PXR. This means results from standard rodent PXR activation studies cannot be reliably extrapolated to predict human induction potential.
Okay, next nuclear receptor, CR, the constitutive androstenedione receptor. Why constitutive?
CR is interesting because it can activate the transcription of its target genes, even in the absence of an identified external ligand binding to it, hence constitutive activity. However, its activity can be further modulated. Certain endogenous steroids, like androstenol and xenobiotics, like the pesticide TCP-OBOP in mice or CITCO in humans, act as agonists, increasing its activity. Other compounds like clotrimazole and meclizine in humans act as inverse agonists, actually suppressing its basal activity.
What genes does CR induce? Does it overlap with PXR?
Yes, there's significant overlap, but also distinct targets. Tau induces many CYPs, including CYP2B6, CYP2C9, and also CYP3A4, like PXR. It also induces various phase 2 enzymes, GSTs, UGTs, ULTs, and drug transporters. Since both PXR and CAR can induce CYP3A4, and they respond to different sets of activators, this contributes to the extremely high inter-individual variability we see in CYP3A4 expression levels.
Does CAR have other roles besides drug metabolism?
Yes, it seems to play broader roles in normal hepatic physiology, including regulating pathways involved in bilirubin degradation, energy metabolism like gluconeogenesis, and controlling liver cell proliferation and regeneration.
And STAR also has species differences in ligand specificity.
Yes, significantly so. For example, the antihistamine meclizine activates mouse KIR, but acts as an inverse agonist inhibitor of human KIR. This again underscores the limitations of using standard rodent models to predict human responses mediated by these nuclear receptors.
What about PPRs, the peroxisome proliferator activated receptors? There are different types.
Yes, there are three main types: PPRA alpha, PPRO beta delta, and PPRO gamma. They primarily regulate lipid metabolism and glucose homeostasis.
Are they all involved in inducing drug metabolism?
PPRA is the main one linked to xenobiotic metabolism. It's the molecular target for the fibrate class of lipid-lowering drugs, like gemfibrozil, phenylfibrate. Activation of PPRA induces enzymes involved in fatty acid oxidation, which helps lower triglycerides. It also induces CYP4 family enzymes, which are involved in oxidizing fatty acids, but could also oxidize some drugs.
What about PPR gamma and beta delta?
PPRA is the target for the thiazolidinone, the TZD class of antidiabetic drugs like rosiglitazone, pioglitazone, which improve insulin sensitivity. According to the source material, PPRO and PPRO do not appear to play a significant role in inducing the major xenobiotic metabolism enzymes like the CYPs, UGTs, etc., unlike PPRA, PXR, and KIR.
And the UGT genes, especially the crucial UGT1A1, are also targets for induction by some of these receptors we've discussed.
Yes, absolutely. UGT1A1 expression, for instance, is known to be a target for induction by ligands activating AHR, PXR, CRF-theta, and also another important transcription factor involved in oxidative stress response called NRF2. Given how abundant UGTs are in the gut and liver, the regulation of their expression by these various nuclear receptors significantly impacts the pharmacokinetics and potential interactions of many drugs, particularly those taken orally that undergo extensive first-pass metabolism.
This deep understanding of metabolism, induction, inhibition, and variability is clearly critical for developing new drugs safely. What role does all this knowledge play specifically in the drug development process itself?
It's absolutely fundamental, really, from the earliest stages right through to clinical use. It's critical for both ensuring a drug candidate has a chance of being effective, it needs to stay in the body long enough, and, perhaps even more importantly, for ensuring its safety.
So what needs to be figured out early on?
You have to identify which enzymes are primarily responsible for metabolizing a new drug candidate as early as possible. Knowing this allows you to start predicting potential drug interactions with existing medications and also anticipate how known genetic variations in those enzymes might affect how different patients will respond.
How is this actually done in preclinical testing? What tools are used?
Researchers use a variety of in vitro systems, meaning experiments done in lab dishes or test tubes, not in whole animals initially. Common tools include preparations of human liver tissue, like liver slices or microsomes, which are fragments of the ER membrane rich in enzymes, cultured human liver cells, hepatocytes, or even purified individual recombinant enzymes, specific CYPs, UGTs, etc., produced using genetic engineering.
And what do they measure in these systems?
They expose the drug candidate to these enzyme systems and analyze what happens. They identify the metabolites that are formed, determine the rate at which the drug is metabolized, and try to pinpoint which specific enzymes are doing the heavy lifting. Is it mainly CYP3A4 or UGT1A1?
Or multiple pathways. And if they find, say, that the drug is almost exclusively metabolized by a single enzyme known to be highly polymorphic, like CYP2D6, or easily inhibited, like CYP3A4, what then?
That raises a potential red flag early in development. Medicinal chemists might then go back to the drawing board and try to modify the drug's chemical structure.
To change how it's metabolized.
Exactly. The goal is often to remove or alter the specific sites on the molecule where these problematic enzymes act, or perhaps introduce sites for metabolism by less problematic enzymes. They try to engineer out metabolic liabilities, particularly avoiding heavy reliance on major CYP oxidation routes before investing heavily in expensive clinical trials.
So maybe an ideal drug, metabolically speaking, might be one that's handled by multiple different enzymes or pathways.
That's often considered a desirable characteristic, yes. If a drug's clearance relies on several different CYPs, or perhaps a mix of CYPs and UGTs, it provides a kind of metabolic robustness. The impact of genetic variation in, or inhibition of, just one of those pathways might be buffered by the activity of the others, leading to more predictable behavior across different patients and situations.
They also specifically test for the potential of the drug candidate to cause induction via those nuclear receptors.
Yes, that's another crucial preclinical check. Using cell-based assays containing human versions of the key nuclear receptors, PXR, CIR, PPRA, researchers can screen drug candidates to see if they activate these receptors.
Why is that important to know?
Well, if your new drug candidate turns out to be a strong PXR activator, for example, you know it's likely to induce CYP3A4 in patients. This immediately predicts potential drug interactions where your new drug could accelerate the metabolism and thus decrease the efficacy of many other existing drugs that are CYP3A4 substrates. Identifying this potential early is vital.
What about using computers to predict metabolism? In silico prediction, does that work?
That's an area of very active research and development using computational modeling. We now have high-resolution crystal structures available for some of the key human CYP enzymes. So computational models attempt to dock the drug candidate molecule into the known structure of the enzyme's active site and use algorithms to predict where on the drug molecule oxidation is most likely to occur.
Sounds promising. Are there limitations?
Yes, significant ones still. The crystal structures give us static snapshots, but enzymes in reality are dynamic and flexible. They can change shape when they bind substrates. Also, the active sites of the promiscuous xenobiotic metabolizing CYPs are often large and somewhat floppy or adaptable, which makes accurately predicting binding modes and reaction sites quite challenging. Similar limitations exist for computationally predicting interactions with the flexible ligand-binding domains of nuclear receptors. It's a helpful tool, but not yet perfectly predictive.
And then there's the standard toxicity testing in animal models.
Yes, that remains a cornerstone of preclinical safety assessment. Acute toxicity studies involving giving escalating doses to rodents over a short period are routine. For drugs intended for long-term human use, like those for chronic diseases, hypertension, diabetes, etc., regulatory agencies require long-term carcinogenicity studies, often lasting 2 years or more in rodents, typically rats and mice.
What do they look for in these long-term studies?
They monitor the animals closely for any signs of toxicity, weight changes, behavioral changes, tumor formation, etc. At the end of the study, extensive necropsy and histopathology, microscopic examination of tissues, are performed on dozens of organs from each animal to look for any drug-related damage or precancerous or cancerous changes. The sheer scale, time, and labor involved in these long-term studies can represent a significant bottleneck in the drug development pipeline.
Are newer technologies like metabolomics helping to potentially streamline some of this toxicity assessment?
Metabolomics is emerging as a potentially powerful, high-throughput approach here, yes. Remember, metabolomics aims for the comprehensive identification and quantification of all the small molecule metabolites present in a biological sample, like urine, blood plasma, or tissue extract, using sensitive techniques like mass spectrometry, LCMS, GCMS, and nuclear magnetic resonance, NMR. You can measure thousands of these small molecules simultaneously.
How does measuring all these small molecules help assess toxicity?
Instead of just waiting to see gross pathological changes in organs, which can take a long time to develop, you can look for earlier, more subtle changes in the overall metabolic profile or fingerprint of the animal or human exposed to the drug. researchers are working to identify specific patterns of metabolite changes in easily accessible biofluids like urine or blood that correlate reliably with toxicity in specific organs, like the liver, kidney, or central nervous system.
So creating metabolic fingerprints or biomarkers that predict toxicity before visible damage occurs.
Exactly. The idea is that these metabolic fingerprints might predict toxicity earlier and potentially more sensitively than traditional methods. This could potentially allow for screening drug toxicity in whole animal systems more efficiently preclinically, perhaps reducing reliance on the most time-consuming aspects of traditional pathology.
Can metabolomics be used in human trials too?
Yes, it's increasingly being applied there as well. It can be used to monitor patients during early clinical trials, potentially identifying metabolic signatures that predict who will respond well to the drug versus who might experience toxicity. It's also used to study the drug's metabolism directly in humans, confirming pathways identified preclinically. And ultimately, the hope is to develop robust metabolic biomarkers that could eventually be used for routine patient monitoring in clinical practice.
And finally, let's touch on the animal models themselves used for preclinical drug development. We already discussed the limitations of standard rodent models due to those species differences, particularly in CYPs and nuclear receptors like PXR.
Right. While traditional rodent models are valuable for high-throughput screening and basic safety assessment, those species differences, especially in the regulation and substrate specificity of the key drug metabolizing CYPs and their regulatory receptors like PXR and COSR, are a significant caveat when trying to predict human pharmacokinetics and interactions. This has spurred the development of more sophisticated, humanized mouse models.
Humanized mice, what exactly are those?
These are mice that have been genetically engineered using techniques like transgenesis or gene replacement so that the endogenous mouse genes for specific human xenobiotic metabolizing enzymes like certain CYPs or UGTs, or their key regulatory receptors like human PXR or CRR, have been knocked out or replaced with the corresponding functional human genes.
So the mouse is expressing the human version of the enzyme or receptor instead of its own.
Precisely. For example, mice have been created that express specific human CYPs in their liver, or that have humanized UGT expression patterns, or whose PXR responds to ligands like human PXR does.
What's the main advantage of using these humanized models?
They can, theoretically, provide a more accurate prediction of a drug candidate's metabolic fate, stability, bioavailability, and induction potential in humans before the drug ever enters phase I clinical trials. Because species differences are generally considered to be less pronounced for some other enzyme families, like FMOs, EHs, most phase 2 enzymes, and for many drug transporters, these humanized models, particularly those targeting CYPs and nuclear receptors, can be quite valuable tools. They might also be useful for longer-term toxicity and carcinogenicity studies, providing a more human-relevant context.
And the material mentions an even more advanced model involving transplanting actual human liver tissue.
Yes, that's another approach creating chimeric mice. This typically involves treating immunocompromised mice, so they don't reject foreign tissue in a way that damages their own liver, and then transplanting human hepatocytes, liver cells. If successful, these human cells can engraft and proliferate, eventually replacing a significant portion of the mouse liver tissue with functional human liver tissue.
So you have a mouse running around with essentially a human liver.
A partially human liver, yes. This model allows studying drug metabolism and toxicity in the context of actual human liver cells functioning within a living organism. However, this model presents its own significant challenges.
Such as?
A major issue is the inherent variability between different batches of chimeric mice, because the enzyme levels and metabolic capacity will depend heavily on the specific donor human liver cells used for transplantation. And those donor cells will reflect the genetics, diet, environmental exposures, and potential underlying diseases of the human donor, introducing significant variability that needs to be accounted for.
So even with the most advanced animal models, careful consideration of their specific strengths, limitations, and the underlying biology is always required.
Absolutely. Mastery of this field really involves understanding not just the enzymes and pathways themselves, but also the nuances and limitations of the various experimental models used to study them.
This has truly been a deep dive, hasn't it? Pulling together so many complex pieces from the simple, fundamental fact of our constant exposure to foreign chemicals to the intricate multi-step processes of phase 1 and phase 2 metabolism. The sheer number and diversity of the enzymes involved CYPs, UGTs, GSTs, and all the others.
Yeah.
The absolutely critical impact of genetic variation and polymorphisms on individual responses, the ever-present potential for metabolic activation leading to toxicity or for drug interactions, and ultimately how all of this incredibly detailed information is utterly essential for developing safe and effective medicines.
It's a remarkable and incredibly sophisticated biological system, far more complex than just simple detoxification.
Yeah.
We've really delved into the versatile but sometimes problematic CYPs, the essential UGTs, and that vital role of UGT1A1 for clearing bilirubin in many drugs, the protective GSTs that shield us from electrophiles, the highly polymorphic NATs dictating acetylator status, and the clinically critical TPMT, where genetic variation can literally have life or death consequences for patients receiving thiopurine drugs.
And what comes through so clearly is that your individual response, your personal reaction to everything you encounter, from the chemicals in your morning coffee to the components of your diet to any medication you might take, is profoundly shaped by this complex, dynamic interplay.
Exactly. Is the interplay between your unique genetic blueprint, especially those variations in the genes encoding these metabolic enzymes and transporters we discussed, and your specific environmental exposures, other drugs, supplements, dietary constituents, even pollutants in the air or water.
The dual nature of metabolism just keeps coming up. It is primarily our essential defense system, allowing elimination and preventing accumulation.
But that same process can sometimes generate reactive metabolites, and its inherent variability, both genetic and environmentally induced, is a major source of unpredictable drug responses, drug interactions, and adverse drug reactions.
And achieving this kind of mastery level understanding of how our bodies handle these chemicals isn't just fascinating biology, is it? It's absolutely crucial for the pharmaceutical industry to even have a chance at developing safer, more effective medicines. And it's fundamental for healthcare professionals trying to predict and manage individual patient responses in the clinic. It really is foundational to good, safe medicine.
It all circles back to the individual patient and their unique metabolic makeup.
Indeed. So as a final thought for you, our listener, as you reflect on everything we've covered today, just consider how your own unique genetic makeup, the specific combination of alleles you inherited for all these CYPs, UGTs, NATs, TPMTs, and others, and how the specific mix of chemicals you encounter in your diet and environment every single day, how all of that is constantly interacting with this incredibly complex, dynamic metabolic network inside you. It's a deeply personalized system. And the future of truly personalized medicine, the ability to tailor treatments precisely and safely for each person, fundamentally hinges on continuing to unravel these intricate individual metabolic stories.